Now, axial filaments, spirochetes bacteria move by the flagellum-like structure, which is called axial filament, which is wrapped around the bacterial cell body and help the movement of these bacteria. That axial filament run lengthwise along the bacterial cell, produced undulating movement or the snake-like movement in the bacteria spirochetes. Their function is similar to the flagella that is in the other flagellated bacteria. Here one can see the axial filament that is wrapped in the whole body of the bacteria, but it is just below the outer membrane. Here also one can appreciate the axial filament that is just below the cell wall and the outer membrane, but it has wrapped the whole body of the bacterium and helps the undulating snake-like movement of this. The capsule, a polysaccharide substance, proteins created by the bacteria around themselves. Thick can be demonstrated by the special technique called Indian ink that is going to stain and to give the clear picture of this capsule. It is up to 10 micrometer thick, mostly consists of polypeptide and polysaccharide. Anthrax bacillus, which has polymerized D-glutamic acid. Sugar component of the capsule vary to produce different serological types. So that's why 84 of the Streptococcus pneumoniae serotypes because of this capsular antigen. Capsule does not have well-ordered structure like cell wall. The capsule is important for many reasons, and it determines virulence of the many bacteria. Capsulated bacteria are more virulent. They are antiphagocytic, that it does not allow the neutrophil to engulf the bacteria, and identification of the bacteria by using antiserum against capsule polysaccharide K antigen and used as antigen in certain vaccine such as the pneumococcal 23, helps in the adherence of the bacteria to the host and protects the cell from the drying, serves as an extra source of nutrition in case of the emergency, so to help to provide the energy. Here one can see the cell wall, and outside of that is the capsule. This is the thick layer of the capsule outside the cell wall, and here one can see the capsule staining, and the bacteria do have the staining of the capsule, and one can see the capsule around the bacteria very easily. Flagella. Some bacteria are motile, have locomotive organs called flagella. These are also called the legs of the bacteria. By that they are able to walk and to move. By flagella, bacteria can also taste their environment, so respond to the specific food poison by chemosensor. So by flagella, bacteria are able to sense whether there is any poison or any chemical which can be dangerous for them. So it moves towards the attractants or away from them. This process is called chemotaxis. These are long whip-like structure, the flagella, embedded in a cell membrane, extend through cell envelope. The long filament act as a propeller, is composed of the proteins called flagell. Here one can see the different parts of the cells, the plates where it takes origin and where it have got the certain rings, and here one can see the filament and filament cap, then there is the hook, and this is the basal body and the motor complex. By virtue of that they are able to move, and here the export substrate which moves inside the flagellum to give its movement. Flagellated bacteria have certain number and location of the flagella. Some bacteria have one, some bacteria have many flagella which are used diagnostically for the bacteria. In some bacteria, flagella are located at one end or the both ends or all over the outer surface of the body of bacteria, seen only in the rods, not in the cocci, or so cocci are the non-motile. Some motile bacteria is like E. coli and the Proteus species cause urinary tract infection. Flagella propel these bacteria up in the urethra and to reach into the bladder. Salmonella species are identified by the use of specific antibodies against flagellar proteins, and Salmonella species are having the flagellar protein. By that we are able to detect the different flagellar proteins. Swarming occurs with some bacteria like Proteus which spread across petri dish, and flagella has three parts: filament, hook, basal body. It is 3 to 20 micrometer long with uniform diameter 12 to 30 nanometer. Whatever the length is there, the thickness is same. Basal body is anchored in the plasma membrane, and the cell wall give rise to a cylindrical protein filament. Flagella attached to the cell by hook, and basal body which consists many rings and rods to give strength to the flagella to be adherent to the cell body and to have the movement through the whip-like structure. Gram negative has two sets of ring and rods, LPS, M rings and rods, as case of E. coli. Here one can see in detail the filament, the hook, the L ring, the P ring, the rod of the flagellum, and these are the different rings. By that it is being C ring, by that it is just anchored and supported in outer membrane, in peptidoglycan layer, in the inner membrane, and inside the cytoplasm. By this structure will give strength to this flagellar body, and flagella do move via the hook and filament. Flagella gram positive have only S and M rings and the rods, the B megatherium. The flagellum moves at its long axis. Arrangement of the flagella, or you can say the classification of the bacteria. Flagellar bacteria, monotrichous or unipolar, have one flagella at one end, and example is Vibrio cholera. Amphitrichous or the bipolar flagella at both ends, and Pseudomonas aeruginosa is an example. Lophotrichous, tuft or many flagella at one end, then cephalotrichous, tuft at the both ends, then peritrichous, which is the flagella all around the bacteria, and the example best is the E. coli. The flagella of peritrichous bacterium must aggregate as a posterior bundle to propel the cell in the forward direction. So that is propeller function of the flagella also. So here one can see atrichous, no flagella; monotrichous, one flagella; amphitrichous, flagella at the both ends; lophotrichous, tuft of the flagella at one end; cephalotrichous, the tuft of the flagella at both ends; and the peritrichous, there is the whole body surface of the bacteria is having the flagellum. The pili or the fimbriae. Pili, Latin, it's the hairs; fimbriae, Latin, is the fingers out. These are the hair-like or finger-like structure outside the cell wall, hair-like filaments, or the projections of the proteins extending from the cell surface. Shorter and straighter than flagella, These are made up of subunits of the protein pilin, so that is why it's called pili, arranged in the helical strands, originates at the cell membrane and extends through the cell wall. Some bacteria have both flagella and the pili, but their functions are different, their structures are different. Escherichia coli, Shigella have 200 per cell of pili. Pili are more rigid than flagella, but smaller than in size than flagella. And these are not involved in the motility, and they just prevent the phagocytosis. Pili are of the many types like K88, K99, F41, etc., with extensive antigenic variation, seen mainly on the gram-negative organism, rarely the gram-positive organism. Example is sometimes seen in Corynebacterium, renal. Pili have got two main important functions. It mediates attachment of the bacteria to the specific receptor on the human cell surface, that is the host cell. Pileated cells help bacterial colonization of the epithelial surface. So the colonization factor is through the pili, and they can adhere to the host cell. Mutants of the Neisseria gonorrhea without pili are non-pathogens. A specialized kind of the pilus, sex pilus, forms attachment between the male and female recipient bacteria during conjugation. I have already shown you the pilus coming out from one bacteria and entering to the other bacteria, which is the recipient. Pili are of two types, short abundant for attachment to the host cell surface and to block adherence to the antibodies. They are going to kill the cell. Pili help in the flotation, increase buoyancy, like pellicle, scum on the water, make froth, so bacteria get more oxygen on surface of the water. They are very long but small in numbers, 1 to 6, known as sex pili or F pilus. So this is the second type of the pili. They can bind to the male specific bacteriophages due to having specific receptors for them. It mediates attachment between the male donor and the female recipient bacteria during sexual conjugation or the bacterial sex. Here one can see the pili. These are the sex pilus from one bacteria to the other to transfer the genetic material. This kind of building can be made only by some super intelligent and genius engineer. Glycocalyx, or the slime layer. It is not a well-defined polysaccharide coating secreted by the many bacteria on the external surface, and in some bacteria it covers surface like a film and allows bacteria to adhere firmly to various structures like skin, prosthetic heart valves, prosthetic hip joints, and the catheters, and it helps the bacteria to adhere them. Also mediates adherence of the certain bacteria like streptococcus to the surface of the teeth, so important in the formation of the plaques, which are the precursor of the dental caries. It prevents phagocytosis as the complement cannot penetrate sugar of the glycocalyx, so it slips out from the complement and cannot be killed. So here one can see the slime layer that is outside the capsule, thick gelatinous jelly-like structure. Bacterial spore or the endospore, these are the modified certain gram-positive rods, especially the Bacillus and the Clostridium species. Genus Bacillus includes agents of the anthrax, and the genus Clostridium includes agents of the tetanus and the botulism. Unusual cell envelope, cell membrane and the outer membrane seen in these kind of bacteria spore cytoplasm is dehydrated and contains calcium diplicate. These bacteria, they make the spores which are present in the bacteria, and that is resistant. They are resistant to the external heat and the harsh environment because of the presence of the dipicolonic acid, a calcium ion chelator found only in the spores. And these spores are highly heat resistant because of its very tough cell wall. And bacterial spores are also called endospore. These are not killed by the boiling, but many disinfectants and dehydration, radiation, and chemicals also cannot destroy them. So, to destroy them, we have to use steam heating under pressure, autoclaving, which is called at 121 degrees centigrade, usually for the 30 minutes, that do kill all these spores. Spores may be found at end of the bacterium, so these are called terminal, or centrally, these are called median or equatorial or subterminal. It may be the shape of the spore is round, oval, or elongate. Once formed, spore has got no metabolic activity and remains dormant for many years. Spores have a thick keratin-like coat, so they survive for many years in the soil. They are formed when bacteria are nutrient deficient, and extreme and adverse condition. Spore formed inside a cell contains bacterial DNA, some cytoplasm, cell membrane, peptidoglycan, and little water. And here one can see the structure of the spore core that is being covered by the coat, which is very resistant. And here one can see the vegetative cell that changes in bad environment to make the spore. Here one can see the cell transform into the spore, and ultimately the spore remains as the part of the cells, and here you can see the spore, and that is a free endospore. Now, upon good environment and the favorable environment, it do change to the vegetative cell and start replicating and can form spore again. So this is a kind of vicious cycle. Though metabolically inactive but contain ribosomes and other essential components, under favorable condition, when the water and the nutrient enter spore, specific enzyme degrade the coat, it germinates to form the vegetative pathogenic bacteria which are capable of replicating. Note, this process is not a means of reproduction. One cell produces one spore that germinates into the one vegetative cell that is capable of reproduction. So here one can see the shape and the position of the bacterial spores, that is oval, and here you can see the spherical, oval subterminal, terminal at one end, and oval terminal, and the different shape at the different position. So vegetative cell transform into the spore, and spore just detach, and when the favorable condition come, the spore germinate into the vegetative cell. So that is the end of the lecture, and I request and hope so that you are going to subscribe this and like this, and please do share it with others. If you have any question or query, you can call me at my number which I have already given. And thank you very much for watching.